Hello students, let us learn the separation techniques like dialysis, ultrafiltration and lyophillization in a simple way. To start with, we'll just go through the dialysis. Dialysis is a separation technique that facilitates the removal of small ions or during purification of macromolecule separation by selective and passive diffusion through a semi-permeable membrane. Cutoff of this semi-permeable membrane can be decided based on the molecular weight of protein of interest. Dialysis is done with buffer, which is usually 200 to 500 times the volume of the sample in which the protein of interest are dissolved. Sample molecules that are larger than the membrane pores are retained on the sample side of the membrane, but small molecules and buffer salts passes freely through the membrane, thus reducing the concentration of those molecules in the sample. Changing the dialysate buffer removes the small molecules that no longer retain in the sample and allows more contaminants to diffuse into the dialysate. In this way, the concentration within the sample can be decreased to acceptable or negligible levels. Dialysis is based on diffusion of molecules in solution higher to lower concentration until equilibrium is reached. Movement of molecule from membrane to liquid will be based on membrane cutoff. Larger molecules cannot pass through the dialysis bag and will be retained inside the chamber. In contrast, small molecules will freely diffuse across the membrane and obtain equilibrium across the entire solution volume, thus effectively reducing the concentration of those small molecules within the sample. If dialysis is allowed to proceed to equilibrium before each change of dialysate buffer, the substance retained by the membrane are purified by a factor equal to the ratio of buffer volume to sample volume. Let us see, for example, when dialysing substance will be decreased 200-fold when equilibrium is attained. Following two additional buffer change of 200 ml each, the contaminant level in the sample will be reduced by a factor of 8 into 10 to the power 6. That is 200 into 200 into 200. If the original sample contained 100 millimole DTT, this would potentially be decreased in the sample to approximately 12.5 nm following the 3 complete cycles. If a further decrease in concentration is desired, the dialysis process can be continued with additional volumes of dialysates. What are the factors that affect the completeness of dialysis? Very importantly, dialysis buffer volume, buffer composition, the number of buffer change, time, temperature, and particle size versus pore size. Substances that are very much smaller than the pore size will reach equilibrium faster than the substance that are only slightly smaller than the pores. Membrane molecular weight cutoff, shortly called as MWCO. MWCO determination for dialysis membrane determines the molecular weight cutoff of the molecule that can diffuse across it. Understanding the signification of molecular weight cutoff, they characterized understanding the significance of molecular weight cutoff determination and how they characterized will allow the selection of best membrane for a particular dialysis application. Proper selection of molecular cutoff will prevent loss of protein of interest and ensure adequate removal of contaminants. How to concentrate protein using dialysis membrane? There are 3 ways of doing it. The first one is the use of hygroscopic substance. Many hygroscopic substances such as PEG, as it is larger molecules than the pore size of the dialysis tube, it will take on water or buffer during the dialysis process due to osmotic pressure. To concentrate the sample, dialysis membrane containing the sample is placed in a small plastic box containing a solution of hygroscopic compound. To avoid contamination of the sample, the hygroscopic compound must be composed of molecules that are or should be high molecular weight. With this setup, concentration occurs upon diffusion of water, that we call it as osmosis, and other small molecules out of the sample and into the hygroscopic solution. Second way of concentrating protein is forced dialysis. In this, vacuum is applied to a sample contained within a dialysis membrane. This effectively pulls water, buffer salts, and other low molecular weight compounds out of the dialysis sample chamber. Third method is diafiltration, which involves pushing samples through a dialysis membrane by a centrifugal force. This is the basis for protein concentrators, which have been popular in the recent years. Let us see how dialysis occurs in human body. In patients with failed kidney, dialysis is often used as a procedure for removing undesirable solids in the blood. During dialysis, concentration of calcium, potassium, and urea dialysate is kept low, thereby facilitating the target solids in the blood to diffuse across the semi-permeable membrane. It is very important to frequently clean dialysate to avoid reaching concentration equilibrium, which could otherwise lead to a rising concentration of unwanted solids in the blood. In another case, solid can also be introduced in the blood. For example, bicarbonate ions. Bicarbonate ions are in high concentration in the dialysate, which diffuses across the membrane. This is done to prevent metabolic acidosis. Second type of separation technique is filtration. Filtration is a common separation method based on a simple principle. Particles smaller than a certain size passes through a porous filter material. The filter traps particles larger than a certain size. The fluid and the particle that passes through filters are called the filtrate or permeate. Depending on the application, the sample type and the scale, filtration systems can vary greatly. Regardless of the simplicity or the complexity of the filtration system, the following 4 components are important. Which are they? The filter itself, a support for the filter, like the funnel, a vessel to receive the filtrate, and a driving force that drives the movement of fluids and particles through the filters. There are different types of filters, but generally they are classified into 3 types according to the size of the particles which they retain. The very first one is a macro filters or general filters, or even it is called as depth filters. These filters are used for the separation of particles in order of 10 micrometer or larger. A coffee filter and a common laboratory paper filter are the examples of microfilters. Next is microfilters. Microfilters are typically used to separate particles whose size range from about 0.1 micrometer to 10 micrometer. Bacteria and whole cells fall in this size range. The third type of filter is ultrafilters. Ultrafilters are used to separate macromolecules on the basis of their molecular weight. For example, large proteins can be separated from smaller ones using ultrafiltrations. Now let us see all these types of filters in bit detail. To start with, macrofiltration. General filters are inexpensive devices which is used to remove relatively large particles above 10 micrometer from a liquid. These filters are made up of materials such as paper, glass fibers, sand, and cloth. General filters do not have pores or holes of a particular size. Rather, they consist of a convoluted granular or fibrous matrix. Particles are trapped both on the filter surface and within the matrix. Because these filters trap particles throughout their depth, they are also called as depth filters. Macrofilters used in the laboratory are usually made up of cellulose or of a glass fibers. In practice, both types may be referred as a filter paper. Glass fiber filters consist of long strands of borosilicate glass that are formed under high heat conditions, and hence, in contrast to filters which are made of paper, glass fiber filters possess faster flow rate, are stable under a wide range of temperature, and are compatible with corrosive chemicals. Manufacturers make a varieties of paper filters and glass fiber filters. These filters vary in the density of their mesh and therefore in the size of particles they tend to trap. Filters that retain smaller particles tend to have slower flow rates than those that trap only larger particles. Next type is microfiltrations. Microfiltrations typically separate particles in the range of 0.1 to 10 micrometer from a liquid or a gas medium. Microfiltration filters are also called membrane filters and are manufactured to have a particular pore size. Particles larger than the rated size are retained on the surface of membrane and the smaller particle passes through. Microfiltration membranes can be manufactured from a varieties of plastics, metals, and ceramic materials such as cellulose, cellulose acetate, cellulose regenerated, cellulose nitrate, Teflon, nylon, polytetrafluoroethylene, PVDF, and glass fibers. What are the applications of these microfiltrations? First, it is widely used to check the sterility of the product. It can be even used to sterile heat-labile compounds such as vitamins, antibiotics. It is frequently used to remove contaminations like bacteria, yeast, and fungi from solutions. And above all, membrane can be used in a quantitative separation or filtration of suspended matter from liquid and gases. HEPA filters, high efficiency particulate air, shortly called HEPA. HEPA filters is used to remove particulates including microorganisms from air. HEPA filters are manufactured to retain particle as small as 0.3 micrometer. Unlike microfilter membranes, HEPA filters are depth filters made of glass microfibers that are formed in a flat sheet. These sheets are then pleated to increase the overall surface area. The pleated sheets are separated and supported by aluminum baffles. HEPA filters have many applications both in the laboratory and in industry. HEPA filters are used in laboratory biological safety hoods to protect from contaminations and or person from exposure to hazardous substance. In animal care facilities, HEPA filters are used on the top of animal cages to protect experimental animal from infections with microorganisms. In industry, HEPA filters may be used to filter the air in entire rooms to protect product from contaminants. The third type of filtration is an ultrafiltration. Ultrafiltration is a type of pressure-driven membrane in which pressure differential across a semi-permeable membrane is used to separate solvent and small solid molecules from solution containing larger molecules. Ultrafiltration usually uses membrane with molecular cutoff of 1 to 100 kilodalton. The differential pressure is achieved by vacuum, pressurized inert gas, or centrifugal forces. Molecular cutoff value is the size of smallest particle that cannot penetrate the membrane. Molecular cutoff values are not absolute because the degree to which a particular solid is retained by an ultrafiltration membrane not entirely depends on its molecular weight. Then what? It is dependent on the shape of solute, its association with water, and its charge also affects its permeability through an ultrafiltration membrane. For example, a membrane is less likely to retain a linear molecule than a coiled spherical molecule of the same molecular weight. In addition, the nature of solvent, its pH ionic strength, and temperature all affect the movement of solid through membrane. By convention, if a membrane is rated to have a molecular cutoff of 10,000, this means that the membrane will retain at least 90% of globular shaped molecule whose molecular weight is 10 kilodalton or greater. The applications of ultrafiltration can be classified as fractionation, concentration, or desalting. Fractionation. It is used for the separation of larger particles from smaller ones. For example, proteins that are significantly different in size can be separated from one another by ultrafiltration. The second one, that is concentration. In this process, solvent is forced through a filter while solute is retained. The initial volume of the sample is thus reduced and the high molecular weight species are concentrated above the filter. This technique is very useful to concentrate sample after protein purification due to the dilution of sample. Before performing gel electrophoresis, the protein must be concentrated because only a very small volume can be applied to the gel. The third application, that is desalting. In this process, low molecular weight salt ions are removed from a sample solution. Ultrafiltration is a simple method to remove salt because they readily penetrate the membrane, thus leaving the solute of interest on the membrane surface. ultrafilters are also ideal for the removal or exchange of sugar, non-aqueous solvents, the separation of protein-bound ligands, the removal of material of a low molecular weight, or the rapid change of ionic or the pH environment. The third separation technique is a lyophillization. Lyophillization is a process which extracts the water from food and other product so that the food or the product remains stable and are easier to store at room temperature. Lyophilization is based on a simple principle of sublimation. What is sublimation? Sublimation is a transition of a substance from solid to vapor state without first passing through an intermediate liquid phase. Lyophilization and freeze drying are terms that are used interchangeably depending on the industry and location where the drying is taking place. Controlled freeze drying keeps the product temperature low enough during the process to avoid change in the dried product appearance and characteristics. It is an excellent method for preserving a wide variety of heat-sensitive materials such as proteins, microbes, pharmaceuticals, tissues, and plasma. The process of lyophilization consists of 3 or 4 steps. First, freezing the food so that the water in the food become ice. Then, place under a vacuum, thus sublimating the ice directly into water vapor. Next, draw off the water vapor. And once the ice is sublimated, the foods are freeze-dried and can be removed from the machine. Lyophilization is useful for the preservation of temperature-sensitive products, for example, biological origin such as enzymes, blood plasma, vaccines, and many more. It is also helpful to provide a practical solution for certain delivery problems. For example, the packaging of constituents that cannot be mixed in the liquid state, but which are solidified in a successive stage and then freeze-dried. It also helps to concentrate the solutes like purified proteins, enzymes, antibodies, vaccines, and also helps to improve storage life and improved marketing of the end product. With this, we end with the 3 important separation techniques. Thank you.